Today we are going to learn about optogenetics. If we divide this term into two, we will get opto means optic, that means related with light, and genetics means genetic engineering. Now, optogenetic approach revolutionized the study of neuronal circuits present in the brain to test the effect of these circuits on behavior. Now, behavior, it could be thirst, movement, feeding and overeating, anxiety, aggression, etc. Now, the whole story of optogenetics began from an unicellular organelles or flagellates, that is green algae called Chlamydomonas reinhardti. Now, this organism responds rapidly to light. Now, see, light is coming into this context. So, this organism can respond quickly to the light, and they show two types of movement. One is called phototaxis. Now, phototaxis means when they move towards the light, and photophobia, that means when they move away from the light. Okay. Now, this organism has a specialized photoreceptive organelle called eye spot, and when scientists actually look at this eye spot, they found that in that photoreceptive organelle there is a photosensitive or light activated channel present. They call it Channelrhodopsin. Now, this protein actually falls under a category of protein called rhodopsin that is present in our retina also, rod cells. This protein is characterized by presence of 7 transmembrane alpha helices like the G protein coupled receptor, and it also contains a photoisomerizable chromophore. Now, this chromophore is very important because this chemical can absorb light. Okay, and by this way this organism actually responds to the light. And this chemical is called all-trans-retinal. So when there is no light, the chemical is called all-trans-retinal, but when they absorb blue light, specifically at 470 nanometer, it converts into 13-cis retinal. So it conversion occurs of this chromophore. Now, as because this chromophore is attached with these 7 transmembrane alpha helices, the body of the protein, so it causes a conformational change. So when transformation or conversion occurs due to the absorption of light, a conformational change occurs into the protein. Okay, and that conformational change helps in the opening of the channel. Now, the channel diameter is approximately 6 angstrom, and it helps when it opens, it helps in the conduction of cations like proton, sodium, potassium, calcium. And this is non-selective cation channels, because in my previous video I have mentioned the channels are very selective, but this type of channels that is present in this Chlamydomonas is non-selective cation channels. Now here comes the role of genetic engineering. Scientists find a very promising approach. Why? Because now they have generated series of designer channelrhodopsins. By changing some amino acids, they can develop different type of channelrhodopsin proteins. Now, that are activated or inactivated by specific wavelengths. As you can see, the natural channelrhodopsin protein that was actually found from this Chlamydomonas reinhardti can only absorb blue light, but by changing the amino acids of this protein, we can make the channelrhodopsin protein to specific wavelengths, any specific wavelengths. Okay, now it not only absorbs blue light, it can absorb yellow, red, or any light by genetic engineering. Also, the intensities of light, and to be selective for a particular, as I have mentioned that this channel is non-selective, but by changing the amino acids of the protein, channelrhodopsin, we can make the channelrhodopsin protein to a selective for a particular cation, and it will help for the generation of action potential. Now, channelrhodopsin protein, another importance of this genetic engineering. See, I have mentioned optogenetics. Opto means optic and light. So this is the light portion of this methods or approach. And genetics, that means genetic engineering. By this way, scientists has made this protein or channelrhodopsins that is specific to wavelengths, intensities of light, and selective for a particular. Now, another benefit of the genetic engineering is that now channelrhodopsin protein can be expressed in virtually any cell type. Now, how this is occur? By using cell type specific promoter. What happens in our body, there are many different type of cells present. Now these cells use different promoter to make different proteins specific for their own usage. Now if we want to express channelrhodopsin protein, suppose in a neuron, what we have to do, we have to express this gene channelrhodopsin to a particular promoter that will express in a neuron. So by genetic engineering, we can express channelrhodopsin in virtually any cell type, like using cell type specific promoter. Example, a subset of neurons present in the neural circuit in a brain, we can express this channelrhodopsin protein. Now by flashing light, we can activate the channel, the channel opens and cations actually enter inside the neuron. So that will depolarize the neuron and create action potential. Now a hole is made in the skull and fiber optic cable actually deliver lights in deeper brain regions. I want to explain one behavioral response, that is thirst response. Now channelrhodopsins are expressed in the subfornical organ, in short SFO, of the hypothalamus. Now in that circuit, neural circuit, there is two types of neurons present. One is excitatory and inhibitory. Now scientists have taken two lines of mice. In one line they express the channelrhodopsin protein in the excitatory neurons, and they took another line where they express the channelrhodopsin protein on inhibitory neurons. Now when they flash light on the excitatory neurons, the neurons get on, or actually action potential occur, and that exhibit thirst even if all the mice are well hydrated. And when they flash light on inhibitory neurons, that also activate the channelrhodopsin protein and it opens and it suppresses thirst. Scientists have performed this optogenetics with different other behavior like movement. In case of aggression, scientists observe that when light is flashed, the mice that is present in the cage, they becomes very aggressive and they attack an inflated hand gloves. And when they turn off the light, they immediately become non-aggressive. So by this way, by the help of optogenetics, we can actually study the neural circuit, because we can generate now action potential in that neural circuit.